To support the investigation of the roles of eotaxin 3 and thymic stromal lymphopoietin TSLP in allergies, asthma, and chronic immune-mediated diseases such as eosinophilic esophagitis, we developed a bioinformatics platform that analyzes the effect of known missense variants collected from public databases on the structure of these proteins. Eotaxin 3 is a chemokine involved in eosinophil recruitment, which plays a key role in allergic inflammation and may contribute to tumor progression. In particular, it is responsible of the CCR3 receptor activation, triggering calcium mobilization and recruitment of eosinophils and basophils to inflamed tissues. TSLP instead is a pleiotropic cytokine produced by epithelial cells. It signals through a receptor complex of the TSLP receptor and the interleukin-7 receptor alpha chain. Two main isoforms exist: the long-form TSLP, which is overexpressed in the airway epithelium of patients with allergic diseases, and the short form, lacking the first 60 residues. Long-form TSLP is considered a master regulator in various physiological and pathological contexts, including asthma, allergic disorders, chronic inflammation, cancer, and autoimmune disease. Targeting TSLP is a promising therapeutic strategy. We, for this work, applied a modeling and analysis pipeline previously established in our laboratory to generate full-length structural models for both proteins and analyzed 105 protein variants for eotaxin 3 and 202 protein variants for TSLP, all derived from known single-point mutations. Because available structures were either incomplete or included regions of low confidence, we generated 10 new models for each protein using modular 9.22, integrating data from the best available experimental structures and AlphaFold prediction. The highest quality models were selected through validation with ProsaWeb, ProCheck, CUMIN, and RMSD, and used as a reference for variance modeling. Structural comparisons between wild-type and mutant proteins were performed using DSSP for secondary structure, HB+ for hydrogen bond analysis, and N-access for solvent accessibility. A custom Python script identified and compared salt bridges. Stability changes were predicted using a consensus approach that included prediction from MaestroWeb, IMS3D, PopMusic, Dynamo2, and Duet. Possible effects on TSLP receptor interactions were examined using PDB-PISA. To share our findings, we developed a dedicated web application enabling users to explore analysis outputs, visualize 3D models, and download structures. For each variant, detailed comparisons with the wild-type protein are provided in side-by-side tables and interactive 3D views. The graph on the left shows the distribution of 105 missense variants along the eotaxin 3 protein sequence, with bar indicated the number of mutations per residue positions. Variants annotated in UniProt as having a moderate impact are marked with a star and circled in the legend. Among the 105 eotaxin 3 variants analyzed, 44 show changes in structural parameters. 6 variants affect conserved cysteines and results in disulfide bond loss. 30 variants show gains or losses in hydrogen bonding with 14 of them also exhibiting reduced stability. As an example of the structural parameters investigated, the figure above, on the left, above the plot, shows the position eotaxin 3 model of wild-type residues affected by mutations, predicted to be destabilizing, along with a visualization of their mutations. In eotaxin 3, we found 22 variants that are less stable than the wild-type protein. Affected residues are shown as magenta ball and stick models. In overlap, the mutated residues are represented by sticks colored as reported in the graphic legend. The same approach has been employed for TSLP. Results show that among the 202 TSP variants analyzed, 121 altered at least one structural parameters. The meter-long form TSLP adopts a 4-helix bundle with an up-up-down-down topology, stabilized by 3 disulfide bonds and connected by loop regions. 4 mutations affected the secondary structure. One, affecting threonine 87, introduces a proline in a loop, causing local distortion, while 3 others target cysteines involved in disulfide bonding. 5 additional mutations affect other cysteines, affecting stability and solvent accessibility without disrupting the overall fold. As with geotaxin 3, hydrogen bonding is the most frequently affected parameter, altered in 96 variants, 18 of which also reduce protein stability. This mutation not only have a strong impact on protein structure, but also impair its interaction with the receptor. In fact, 25 mutations disrupt the interaction with TSLP receptor. Here we show an example. Notably, mutation at arginine 115 and 153 break K-salt bridge with aspartate 92 on cytokine receptor-like factor 2, while mutation at serine 45 causes the loss of an important H bond with tyrosine 212 of interleukin 7. The goal of our platform is to provide this and similar information, emphasizing the importance of elucidating the structural characteristic and genetic variability of this protein, providing support for the development of more precise therapeutic strategies.